Why does grass appear green to our eyes? Again, why do all things around us appear in a variety of colors? We see the light reflected from things when we recognize color. Shall we try to test the reflectance spectrum to see how the colors in printed material or on a color TV are reflected and enter our eyes? The materials you will prepare for the experiments are a reference reflector, the SV2100, and red, orange, yellow, green, blue, indigo, and violet colored sheets. When you measure a reflectance spectrum, you must do the experiments after turning the light source part of the SV2100 to a 90-degree angle as in the picture. First, close the shutter of the light source and measure the 0 point by clicking the dark button of the program. Next, turn on the light source. After opening all of the shutters, insert the reflector into the groove formed just 45 degrees diagonally in the sample holder and measure the maximum value by clicking the reference button of the program. At this time, control the shutter and integration time for the maximum intensity of the program to be less than 16,000. Here, when you measure dark, reference, and sample, you will carry out the experiments with the shutter and integration time kept in the same condition. Now, let's try to measure the sample you had prepared. After you pull the reflector out of the cuvette holder, insert the prepared reflecting red sheet into the groove of the holder, forming a 45-degree angle diagonally, and then measure the spectrum. You can confirm the degree of reflectance from the measured spectrum by using the reflectance button in the program. After measuring the spectrum with the orange, yellow, green, blue, indigo, and violet sheets that you had prepared, put it in the diagonal groove of the holder in order. Let's measure at the reflectance mode and organize the results. The x-axis of the measured spectrum shows the wavelengths of the light. 400 to 430 nanometers means violet, 430 to 450 nanometers indigo, 450 to 500 nanometers blue, 500 to 570 nanometers green, 570 to 590 nanometers yellow, 590 to 610 nanometers orange, and 610 to 700 nanometers red. If you observe the reflectance spectrum, the reflectance spectrum of a red sheet reflects the wavelengths between 610 and 780 nanometers and absorbs the rest of the color wavelengths. The reflectance spectrum of a yellow sheet reflects the wavelengths between 570 and 780 nanometers of yellow and absorbs the rest of the region. The green sheet reflects wavelengths between 500 and 570 nanometers of green, and a blue sheet reflects the wavelengths in the region between 450 and 500 nanometers of blue. You will see that the spectra of orange, indigo, and violet sheets reflect the wavelengths equivalent to each color strongly in the same way, and absorb strongly in the rest of the region. As a result, we can understand that the color which our eyes recognize is a reflected light. Our eyes contain two kinds of vision cells, called cones and rods. The cones sense color under bright sunshine in the daytime, and the rods sense objects or images as black and white pictures in the nighttime or under low illumination. Such cones are divided into red, green, and blue cells, with each cell identifying the color according to the stimulating strength of light that each cell receives. When a certain material reflects all the wavelengths of light, the stimulating strengths that three kinds of cone cells receive show a high value. As a result, the material appears white. In an opposite case, because the stimulating strength of three cells show a low value, the material appears black.